Our topic this week is DNA. We need to get a good understanding of both the structure and the function of this molecule to help us as we move forward thinking about genetics, biotechnology, and a whole variety of other topics. So I want to walk you through the key elements of DNA structure. And bear in mind that this will be true of DNA from prokaryotes like E. coli, to plants, to fungi, to you and I. We're all using this one important molecule to carry and pass on our genetic information. So here it is. Not really, but this will help us think about it. DNA is a double helix. You might have heard that term. It might be easier, though, to picture a spiral staircase. This is the structure we're going to be after in building our understanding of DNA. And when I think of this staircase structure, I like to first imagine a ladder with rungs. So we're going to build a ladder together, and then in your head you can coil and twist that ladder till you get a double helix shape. So making up the sides of each ladder are a very tightly bound, repeating pattern of sugars, phosphates, sugars, phosphates, sugars, phosphates. So in my model here, that's what I'm seeing with these light brown side rails or bands here. These are just alternating sugar, phosphate, sugar, phosphate, sugar, phosphate groups, all covalently bonded to one another very, very tightly. In addition, hanging off each one of the sugars in my sugar-phosphate backbone is what we call a base, a nucleotide, and you've probably heard these referred to as A's, T's, C's, and G's. One of these bases, an A, T, C, or a G, is covalently attached to each sugar in that backbone. So if I'm thinking about my ladder analogy again, my two side rails are those phosphate, sugar, tight, covalently linked molecules there. Hanging off each sugar, if you can picture my thumbs here, are nucleotides, an A, T, C, or G, also covalently bounded to that sugar. So these are really rigid, stable structures. This is why we can get DNA out of mummies and dinosaur bones. It's a tight, heat-resistant structure. We need to glue our ladder together, right, these two halves though, and to do that we're going to use a much weaker intermolecular force, our hydrogen bonds. So these two bases are going to stick to one another because of relatively weak hydrogen bonds. So they're doing that up and down the length of the molecule, making the rungs in my ladder, or I might think of them as the stairs in my spiral staircase. So nice, two strongly covalently bonded pieces of DNA here, held together by hydrogen bonds. So there's one big idea for us. The second big idea I want to point out is that you can then separate and reattach these two halves of a DNA molecule very, very easily. We do this in the lab with a little bit of heat, and our cells are doing it constantly for DNA replication. This is possible because the DNA helix always comes back together in a very predictable pattern due to what we call complementary base pairing. This means that A's can only form stable hydrogen bonds with T's, and C's can only ever form stable hydrogen bonds with G's. So if I know a sequence of DNA reads A, C, G, the other half of that molecule is going to have to read T, G, C. Because again, A always pairs with T and C always pairs with G. This was a critical piece of the Watson and Crick model of DNA, and they realized very quickly that if you understand complementary base pairing, then when I separate the two halves of a DNA molecule, I can use each half as a template to rebuild an identical molecule. This is what your cells are doing every day before they go through mitosis or meiosis as they replicate the genome in the nucleus. Simple DNA replication.